Baltic Agro talveseminar. Silo ja sööda kuumenemise ennetamine. Kogemused Soomest.
Hea silotootja, mina olen Tiina Meikar. Järgneva ettekande teemaks on, kuidas ennetada silo ja sööda kuumenemist. Antud teemat tutvustab Arja Seppälä. Arja on igapäevaselt Soome AIV-toodete tehniline konsulent, kes omab silomaailmas pikaajalist kogemust nii teadlasena kui ka praktikuna. Millest täna juttu tuleb? Mis juhtub silo toiteväärtusega kuumenemise käigus? Millised on riskifaktorid? Miks ja mis toimub, kui söödasegu söödalaval kuumeneb? Lisaks tutvustab Arja ka AIV tooteportfelli. Siinkohal ma annangi sõna Arjale.
I want to tell you good news that good aerobic stability can be achieved together with excellent fermentation quality when using right silage additives. So, about the silage heating, first let's start to think about what actually happens after silo opening. So when silo is opened, microbes get oxygen. And when they get oxygen, they are able to consume lactic acid and sugars. They cannot consume lactic acid in anaerobic conditions, but especially some yeast species are able to consume lactic acid when there is oxygen present. And typically, the higher the yeast, the count, the number of live yeasts, the higher the number of yeast is, the quicker is the heating after silo opening. And what happens after that is temperature starts to increase, acidity will be reduced. So because microbes consume lactic acid, and the pH start to rise. As well, moisture will be created during this process, and silage will get more wet. And let's think about what happens to silage nutritive value during this heating process. So first of all, sugars and lactic acid will be consumed, and they may actually be totally lost. And because they are fully digestible nutrients, that's a bad thing. They cover more than 10% of dry matter, and they cover more than 16% of digestible nutrients in the silage. So definitely we don't want to lose those. And let's further think about the consequences of silage heating and TMR heating. So there's loss of nutritive value, reduced digestibility of the feed because we lost those most digestible nutrients. Furthermore, feed intake will be reduced as well. Milk production and meat production will be reduced. And often that is also detected by increased feed sorting by the animals. Of course, extra work will be caused when this spoiled feed has to be removed. Also, the amount of leftovers is often increased. So that's the amount of leftovers is often the first signal detected by farmers, so that they realize that now there's something wrong. And finally, health problems may occur due to this unbalanced feeding, especially when animals start to sort, and though they are not consuming silage, they tend to select only the concentrate. So that is finally causing unbalanced feeding, and of course that is leading to health problems, and cows in early lactation are most sensitive to these kind of things. Here's some example of from Finland. There was the advisory services. ProAgria was following some farms and about the measuring silage temperatures, and they noticed that when silage temperature was high, so average temperature 33 degrees, milk yield dropped by 1.7 kilogram energy corrected milk per cow per day. More concentrates concentrates were fed to prevent further drop in milk production, and silage intake was reduced by 2.6 kilogram dry matter per day per cow. So quite extreme changes. So definitely these things should be avoided. And a list of risk factors which may cause silage heating. This is quite long list, and but basic things are to understand that we should by all the means prevent aerobic spoiling microbes. We should prevent that porosity so that oxygen should not penetrate within silage, and we should use proper silage additive. And let's first talk about aerobic spoiling microbes. What are the factors that may increase their numbers? So it's prolonged pre-wilting in bad weather condition may cause their numbers to rise. Soil contamination or dead plant material often has high number of yeast and molds, and as well as bacteria. Any holes in covering plastics give oxygen to yeast, and then they will survive in the silage. As well, sometimes I have seen that plastics are not firmly attached to the silage. So if the wind is, for example, moving the plastics, then there will be problems because the microbes get the oxygen too early. Or if silo is open too early, so that aerobic microbes are still alive. So that may also cause problems. And the porosity, high diameter often makes silage difficult to compact. Whole crop cereal silages like barley, for example, are more difficult to compact than grass silage. Insufficient compaction generally. Long shop length make it also difficult to compact. And the one thing is method how silage is removed from the silo. So there should be used some cutting device and not just a front loader. And slow feed out rate should be sufficient so that consumption is high enough. And if silo is too wide, then there is also a risk that consumption is too low per day. And then there's also factors related to silage additive. Wrong type of silage additive that can't improve aerobic stability. For example, it's good to know that lactic acid alone can't prevent heating because lactic acid is actually feed for the yeasts. It's not preventing them. It's food for them. So they are consuming lactic acid. So if there's only lactic acid produced during ensiling, there's nothing that would prevent yeasts. And furthermore, sometimes the additive is right one, but the application level is too low, so that then there's not enough effective substances to prevent the heating. So this is the long list, but good to check if you have any issues with heating, so that at least you could improve next year the actions. And I will tell you that AIV silage additive is effective to improve aerobic stability. So in many ensiling trials, we have seen that during ensiling, when AIV is used as silage additive, it is killing the yeasts during ensiling. And finally, when silo is opened, there's good stability in the silage. So also you can see that in high dry matter silages having dry matter 38%, AIV has been able to improve the stability substantially. So this is good example how you can by using a right silage additive, you can improve stability by more than 1 week or even 2 weeks. So this is a really big value for your silo management, so that you don't need to remove the spoiled silage. And then some tips what to do what is the first aid when silage is heating? Here is the photo from one farmer who had a heating case. The silage was harvested using a forage wagon, and the shop length was quite long, and the additive was not used in proper manner, so there was not enough additive, or it was not evenly distributed. So they had a real heating problem. It didn't start immediately after silo opening, but only 2 weeks after silo opening it was detectable. So because of course, first when you open the silo, the yeast starts growing, but after 2 weeks there is already high population, and air is progressing. Air is entering into the silo and going further into silo. So that's why the heating problems normally is detected something like 2 or 3 weeks after silo opening. And in this case, the farmer was adding some extra weight in top of the silo to keep the plastics on top, because there was not enough weight and the compaction has not been sufficient. And further, some tips what to do. So it's good to remove the silage that is already heated, and if possible, consume the silage quicker. And if that's not possible, try to consume, for example, 30 centimeter from the whole silo weight from each day, instead of consuming 60 centimeters every second day. This way, you can peel the spoiling surface away more often. Use Eastman Stabilizer TMR to prevent TMR heating, and check what to do better next season. So it's always when you have a problems, it's good to consider how to improve things next season. And then something what you can do with the total mixed ratio. So often, if you have silo heating in the silo, you have also heating in the TMR. Or if you have heating in the TMR, you probably have the reason is that your silage is heating. So these things are really strongly linked to each other. So why TMR stabilization is needed? It can be that there's increased amount of refusals, intake is less than expected or below optimum, there's increased sorting. These all are small signals that there's something wrong, and you could improve by using TMR stabilizer. Or it can be simply that you detect heating in either TMR or some ingredients of TMR. If water is added into TMR to prevent sorting, then it's really recommendable to use TMR stabilizer. You can actually add it into the water and add it together with the water into your TMR. So that therefore, because the water is microbes need some moisture to grow, and if there is water added into TMR, then the risk for heating is even increased. So therefore, it's good to remember that whenever you add water into TMR, please always use TMR stabilizer. And then it's also, of course, high ambient temperature. So microbes enjoy the warm summer days and grow more rapidly then. And TMR stabilization is quite easy. It's so that actually you see the immediate effect immediately within the same day when you have started using the stabilizer. And effect on animal intake and animal responses you will see also the same day or soon afterwards, so the following days. And application level, you can easily adjust it based on your on-farm experiences. So normally my recommendation that let's start with 3 liters per ton, and if that's not enough, you can increase the application to 5 liters per ton. And if you think that that's already enough, you can also reduce this afterwards. So you can always adjust the application level based on your own experiences, and you will see the effects immediately the same day. And our product, which we recommend to you, is Eastman Stabilizer TMR. We have had good trial results. Here is one example from Finland, conducted in Luke the research agency what we have in Finland. So we see that Eastman Stabilizer TMR did really well in this trial, outperformed many of the other products. It's a liquid product which ensures even distribution and immediate effect. It's non-ADR, safe to use without pungent smell. So, and product composition ensures wide efficacy against several types of spoiling microorganisms. That's the reason why there is formic, propionic acid, and benzoate in that product. There's also caramel color to make it easier to use, and propylene glycol to reduce corrosivity, as well as sodium formate to reduce corrosivity. Now it's time to hear about AIV 2000 plus NA. I will tell you about the composition and benefits of that product. And AIV 2000 plus NA is an silage additive that can be used to avoid any problems related to silage fermentation quality or silage heating. Let's look at the silage ensiling process. We can divide it into three phases. So there's pre-wilting phase, then there's ensiling phase that is totally anaerobic, and feed-out phase that is again aerobic. And during pre-wilting, oxygen is present, aerobic microorganisms will grow, and pH is still high, about 6 or so. But when silo is closed, oxygen is rapidly depleted, and aerobic microorganisms will die. pH will drop when some acid is added into silage, or when lactic acid is produced. So the fermentation phase typically takes something 14 days, and during that time fermentation takes place, and lactic acid and probably also some acetic acid is produced. After that, there's anaerobic stable phase, when silage should be relatively stable, so minimal changes. And when silo is opened, oxygen is again present, and silage may start spoiling, so that it may start heating and pH may start rising again. And what does AIV do here during this process? So in the very beginning of ensiling, AIV is ensuring that pH will drop rapidly. It's immediately dropping because formic acid is a relatively strong acid. Furthermore, during ensiling phase, it's restricting fermentation so that there will not be too much lactic acid and no too much acetic acid in the silage, or no butyric acid. Furthermore, AIV is helping to improve aerobic stability, so it's killing yeast during ensiling. So in the feed-out phase, silage will be more stable without problems related to heating. Let's look at the composition of AIV 2000 plus. So main component is formic acid. It's causing the effective rapid drop of pH in the beginning of ensiling. So that is the really important thing. And formic acid molecule as such is preventing harmful bacteria and yeasts. And then there is sodium formate, which is the salt of formic acid. It is there to reduce corrosivity, but it's also contributing to microbial control. So it's also also controlling bacteria and yeasts. There's also propionic acid and sodium benzoate in AIV 2000 plus. Let's talk about them a little bit later, but now let's talk first about the pH drop. So dropping the pH is really important. First of all, it's preventing butyric acid fermentation in silo. It's also preventing protein degradation. And finally, when pH is close to 4, it's also preventing lactic acid bacteria and also restricting the total amount of fermentation. So it's necessary that pH is reduced during ensiling, and that's the so-called AIV principle. There's about thinking about the organic acids. I would like to compare formic acid with lactic acid and acetic acid and tell you some features about those acids. Let's look at the formula. So you can see that formic acid molecule is really small, while lactic acid is about double in size, and acetic acid as well is much bigger than formic acid. So formic acid is unique nature because it's so small molecule and it has relatively low pKa value. This pKa value is related to efficacy of the acid. So it's determining in which pH the formic acid molecule or any other organic acid is releasing its proton and making the solution acidic. So these two things, molecular size and pKa value, are affecting how much acid is needed so to a certain pH drop. So let's say that we need to drop the pH to 5 and we would need formic acid 1 kilo, for example. So lactic acid is needed about a double amount and acetic acid as well about a double amount to achieve the same pH. But when we go further down, when we want to reach pH 4, the difference between these three acids comes even more bigger. So lactic acid you need nearly 3 times more, more than 3 times more, and acetic acid you need nearly 9 times more because acetic acid has so high pKa value compared to lactic acid and formic acid. And lactic acid has a bigger molecule, and that is the main reason why that is needed much more than formic acid. So really, these pictures emphasize how unique formic acid molecule is and why it is used as silage additive. And then there's propionic in the AIV product. So on top of formic acid, there's propionic acid and sodium benzoate. So propionic acid is there to prevent molds and yeasts, and it is as well glucogenic energy source for ruminants. And its pKa value is 4.88, which means that most of the propionic acid is undissociated in silage, which means that it's highly effective against microbes. Sodium benzoate, in the other hand, it also has synergism with other constituents of this AIV product, and it's also preventing molds and yeasts. So it still really has its role in this product. And furthermore, I want to tell you about the synergism between formic acid and propionic acid. So when formic acid is applied into forage, it acts rapidly dropping the pH. Because the pH of the grass it's originally about 6, and when formic acid applied there, formic acid is releasing the proton, the hydrogen ion, which is causing the pH drop. And so the pH will be now in the silage is about 4.2 or so, and which is much lower than the pKa value of propionic acid. And due to this difference between these organic acids, propionic acid is actually in the undissociated form, which can penetrate the cell wall of microbes. So it is because these two organic acids have so different pKa values, they are acting differently, and formic acid is making propionic acid more active against microbes. So it is quite nice that silage pH is between the pKa values of these organic acids. And I want to remind you also that organic acids are naturally present in living cells. So formic acid is found in ants as well as in some rumen microbes. Propionic acid is produced in rumen microbes, and it's important glucogenic energy source for ruminants. And sorbic and benzoic acids are also found naturally in the many berries, for example, lingonberries. So this kind of you can think that they are natural compounds such a way that animals, you can safely feed them to your animals. And why to use AIV 2000 plus NA as silage additive? We have listed here the main reasons: to prevent malfermentation during ensiling, to reduce risk for silage heating during feed out, to maximize feed intake and milk production, and to maximize milk fat and protein content. And when to use AIV 2000 plus NA? When you want that your silage has the best possible quality, when you have difficult crops to ensile, legumes or wet timothy for example, and when you want to be better prepared for bad weather and to wet crop. Something to remember and pay attention is that you need to succeed with applying silage additive. And here are some things to remember and to check. It's always good to use drive over scale to measure the harvested forage load. And at the same time, follow additive consumption so that you are all the time aware how many liters per ton is really consumed. Check and test capacity of your applicator, including pump, tubes, and nozzles. The whole system, so the capacity of the whole system is determining the capacity of your applicator. And check that the size of the nozzles and location of the nozzles are optimal. And the pumping one crucial thing is the pump. Is the capacity of the pump high enough for high yield? When you have high grass yield, you surely want to harvest it quickly. So then it's crucial that your pump is has high efficacy so that it's able to apply the needed amount of additive. The pumping capacity required by a forage harvester is typically approximately 10 to 30 liters per minute. And we cooperated with Hapuva, which is a pump, which is selling pumps for us harvesters and other harvesting equipment, and you may look more information on their web pages. I want to show you also some trial results. This was made 2019 in the Helsinki area, so really in the southern Finland. Maize ensiling trial. There were two harvesting times, so two dry matter concentrations. The lower one is 26% and the higher one is 31%. Maize is many times considered quite easy to ensile, and it's true partially, but often I have seen maize silages which have high acetic acid concentration or poor aerobic stability. These two problems are common with maize silage, and these are further avoiding, and they can be avoided. And among these results, I want to show you that acetic acid concentration could be restricted or could be controlled by using AIV 2000 plus NA as an ensiling. As you see with the lower dry matter concentration, when control or no additive or some competitor products were used, there is a little bit elevation in the acetic acid concentration. And if the ensiling would be, if the maize silage would have been in the silo longer time, most probably there might have been more acetic acid produced in it. But the lowest level was measured with AIV 2000 plus. So it's capable of preventing acetic acid in wet maize silage. And among the other parameters, proteolysis it was reduced by AIV 2000 plus, so the lowest ammonia concentration in that silage, and also the total amount of fermentation acids was lowest when AIV 2000 plus NA was used. And the clear effect in aerobic stability can be seen, so that AIV 2000 plus was clearly improving aerobic stability. So this means that it was preventing heating during the feed out. So whenever you have some problems with maize silage, consider using AIV 2000 plus NA. Then grass ensiling trial. this was conducted in Finland, Natural Resources Institute Finland, and it was grass that was ensiled. By the fermentation quality, all of these silages were quite good. Even the control without additive was relatively good quality, low ammonia concentration, etc. But the biggest effect is seen in aerobic stability. The aerobic stability of no additive treated and biological treated silages was poor, while it was extremely good with AIV 2000 plus treated silage. So sometimes the biggest value is really seen into improved aerobic stability. Here is one more trial with alfalfa lucerne. This was really wet. The ensiling trial was conducted in Germany, late autumn 2016. AIV was applied 5 liters per ton, and we could say that it was not, we would recommend using a little bit more because this was so difficult material to ensile. So without any additive or with inoculant, the ammonia concentrations were extremely high and volatile fatty acids were extremely high. You can see 150 grams per kilogram dry matter of volatile fatty acids. It's I don't know if that kind of silage cannot be fed to animals actually, because it's so bad. But with the lactic acid, with the AIV, it was possible to prevent those wrong type of fermentation. And with the strongest AIV product, it was even possible to spare some sugars. But really, if you need to ensile really wet lucerne, it's better to use 6 to 7 liters per ton of AIV because it's so difficult material to ensile. But this kind of, if you think about what are the effects in milk production in this kind of situation, more milk could be expected 1 to 1.7 kilogram energy corrected milk per day, and milk fat and protein concentrations would be substantially increased if you have proper silage. And here with the grass silage, here we can see that milk production was increased by using AIV 0.7 kilogram more milk compared to lactic acid bacteria treatment, and 0.9 kilogram more milk compared to no additive silage. And that was because there was more sugars in the silage that was made with AIV and less fermentation products, so that cows prefer to eat the silage which is restrictively fermented, so that the amount of acids, fermentation acids like lactic acid and acetic acid is small in the silage. So finally, some summary. Effects of AIV in ensiling. So the key thing is the rapid drop of the pH in the very beginning, and which is preventing respiration and proteolysis, as well as breaking cell walls and improving consolidation of the silage and inhibiting undesirable microbes. The formic acid molecule itself is helping during fermentation phase to restrict fermentation. And during aerobic phase, AIV silage has low number of aerobic microbes which prevent heating, improve aerobic stability, and that's why you get the losses will be minimal. And finally, restricted fermentation is reflected as better microbial protein synthesis in rumen and higher intake. And finally, more milk fat, and milk protein will be produced. And the benefits for the farmer with using AIV is less weather-dependent harvesting. You can also harvest good silage, although the weather is not optimal for prevailing. You will have minimal losses, higher feed intake and higher milk yield, and better milk quality. Thank you for your attention for listening this lecture. Thank you.
Mida kokkuvõtteks meelde jätta AIV tootja soovitustest? Silo ja sööda kuumenemine toob kaasa toiteväärtuse ja söömuse languse. Söödasegu kuumenemise söödalaval põhjustab enamasti aeroobselt ebastabiilne silo. Kuumenemist on võimalik vältida. Stabilizer on abiks kuumeneva sööda stabiliseerimisel. Kasuta AIV tooteid nii märja kui kuiva rohumassi korral. Oluline on õige doseerimisnorm. Kontrolli pumba võimsust, et doseerimine oleks piisav. Kasutades AIV tooteid on tulemuseks piiratud fermentatsiooniga, hea toiteväärtusega, aeroobselt stabiilne silo. Lihtne! Aitäh teile! Rohkem infot Baltic Agro kodulehelt ja blogist. Edu ja õnnestumisi!